All right, so today's lecture we're going to start talking about particle separations. The previous lectures in the module were all about molecular separations. So molecules are assumed to be dissolved in the solution, right? Dissolved in the medium or buffer, right? So we talked about chromatography, we talked about electrophoresis, we talked about the H filter. So these are methods that were used to separate molecules based on the concept of diffusion, in the case of the H filter, on the basis of charge and size, if we're talking about electrophoresis. And if we're talking about chromatography, you can separate based on a whole bunch of different properties. Size could be one of them. Surface properties like hydrophobicity, hydrophilicity could be another one of them. Charge could be another one of them. It depends on, like, whatever you want to separate based on depends on, that'll determine the type of column that you purchase, HPLC column. You can purchase different ones for different reasons. Now, to contrast that, today's lecture is about particle separations. Particle separations are actual particulates that are not dissolved in the sample, but rather they're little particles that are suspended in the sample. So these samples would be referred to as suspensions because you have particles suspended in the medium. So there's a lot of different applications of why we'd want to do particle sorting. We've talked so much about the blood, so you know that this is one of those things, one of those examples. In a blood sample, we have like red blood cells, we have white blood cells, we have platelets. You have different kinds of you could have foreign cells in them, you could have bacteria in them, you could have cancerous versus non-cancerous cells. In fact, one of the big areas in this field right now is the ability to capture and sort circulating tumor cells in the blood. It turns out that circulating tumor cells have slightly different properties than normal cells. Okay. Some of the most recent stuff that we're not going to talk about in this module have found that circulating tumor cells are more rigid. They're less squishy than normal cells are. Some of like Dino DiCarlo's work at UCLA, for example, looks at analyzing the deformability of cells and being able to sort cells based on those parameters. There's some work that was published earlier, and this is well known now, that cancer cells have different size, a slightly different size than normal cells do. They can be slightly larger. So if you could separate cells based on size, and you could do that with very high resolution, the idea would be you'd be able to separate cancer cells from non-cancerous ones. Most recently, some of the things that have actually been shown to work is cancer cells have different surface markers than regular cells do. So we've talked a little bit about this before, but I'm just going to put up a really rough diagram here. A cell might have different surface proteins on it. You know, protein has these. A cell has these different proteins on the surface. I'm just going to draw them like this. So this would be the cell membrane. This would be a membrane protein. These membrane proteins have functions in the cells. We're not going to go into all the biology of that. But different cell types have different proteins. You know, this is the inside of the cell, it's the nucleus, and the cytoplasm is over here. So it turns out cancer cells express different types of membrane proteins. These are also called surface markers. And you can create antibodies. You can create these surfaces, or like pillar-like structures that present these antibody groups, and these antibody groups will capture the cells as they pass by those surfaces. So imagine having an array of these pillars, and the protein and the cell is being pushed through that pillar array. Eventually, the cell will bump into one of those pillars, and it will be captured by these antibodies that have been functionalized on the wall. Okay. So the point I'm trying to make here is that there's a lot of interest right now in looking at the different types of particles in the blood, whether there's red blood cells, white blood cells, cancerous, non-cancerous, platelets, what have you. If you could sort by size, by deformability, by surface properties, then you can count each type of cell. One example of where cell counting is very important is in HIV diagnostics. CD4 is a certain cell type that is indicative of whether a person has HIV or not. More conventional type things, if you go to a doctor's office, you often get red blood cell counts, white blood cell counts, platelet counts, and so on. I mentioned metastatic cells, circulating tumor cells. They're starting to be able to count those as well. The number of circulating tumor cells that you may have in your blood is indicative of what stage of cancer that you may have at the particular time. By analyzing the circulating tumor cells, you can actually find out where the cancer originated. Okay, all these techniques require methods to capture particles or capture cells in a liquid, to be able to sort them based on different properties, and so on. So besides blood analysis, you can also imagine environmental analysis, bacteria sizing, other live organisms. In a typical beach water sample, you'll have many different kinds of organisms, ranging from single cell organisms to bacteria. They could have sizes ranging from 2 micrometers all the way up to several hundred micrometers. If you could take that sample and separate out all the different organisms by size, you could sort of classify and count how many bacteria you have, how many paramecium you have, and so on. You see, and this is very useful for people who are monitoring invasive species in the environment. It's important for looking at when a certain beach, for example, becomes unsafe due to high bacteria levels. So there's a lot of different types of applications, both in biology and life sciences, and also in environmental analysis for particle sorting. Cells are basically particles in solution. Another big application is flow cytometry, and I'm mentioning this because this is a lot of the cell sorting and focusing techniques that we're going to talk about later in this module are ultimately applied to flow cytometry. Flow cytometry is a method for counting and differentiating different cell types. So I was mentioning in the previous examples, we want to sort cells by type, and then we'd also want to be able to count each one. So this technology or this concept has been around for a long time. The field is called flow cytometry. It's a method for counting and differentiating different cell types, and the idea is that cells will flow single file through a window where they are detected by a laser. All right, so imagine that you have a sample, and that sample contains cells in suspension of cells. In flow cytometry, that sample is combined with 2 other flows of liquid, which is called the hydrodynamic focusing, or it's also called sheath flow. Now, this is similar to the concept of laminar flows that we saw earlier. This stream that you see here is combined with the 2 streams on the outside, and they form these parallel streams through the length of the channel. Now, 2 things happen. First of all, this sample, which had a concentrated agglomerate of cells, those cells actually get spread out in time, or spread out along the axial direction. This technique is referred to hydrodynamic focusing because the 2 flows along the outside, or rather it's coaxial. It's going around the inner tube. The 2 flows, the flows around it, it squeezes together. It focuses the flow on the inside. This is referred to as hydrodynamic focusing. So I'm just jumping ahead here. We'll come back to this slide. If you have one fluid flowing through a channel like this, and then you have 2 fluids kind of squeezing it from both sides, that middle stream actually gets squeezed together into a thin stream like this. Now, if you have particles in that flow, the particles are going to get squeezed into a single file line, and the individual particles will get spread out in time. This is exactly what you need for a cytometer. You need the particles to be single file. They're not overlapping with one another, because then you'd end up counting the same. You can end up counting 2 particles as one. You don't want them aggregating together. You want them single file, and you want them spaced out. So as they're shooting by the detection region, the detector can count each one. So you can control the flow rates of the focusing streams, and that will ultimately determine how how thin or how focused the middle stream is. So this is the focusing portion. Once you get past the focusing portion, there's the detector portion. The detector consists of a laser which is focused into the center of the microchannel. Okay, the laser is focused just on the specific spot where these cells are expected to travel through, the flow line. Now, these cells are fluorescently tagged. We haven't talked much about fluorescence yet, but the basic idea behind it is that the cells will glow a certain color if it's excited with a higher energy light. For example, you have, let's say, a red fluorescent stain. So we've seen examples of fluorescence because if you have like a glow-in-the-dark anything, that's usually some type of fluorescence. A very common fluorophore is fluorescein, which glows green. Again, a lot of times when you see the pictures of cells in journals, the cells are glowing green and red. That's because the cells have been stained with the fluorescent agent. So in this case, the cells are stained with a fluorescent dye, and that causes the cell to glow a certain color when it is excited. Now, in this particular example, this diagram, they're showing that the cells are they have a red fluorescence that's emitted. You shine green light at the cells. Okay, the green light is absorbed by the fluorophore, the fluorescent dye. It absorbs green light, but it gives off red light. So you can detect the red light emitted from the cells and count the number of cells that were stained red. Now, another important thing here in flow cytometry is this the concept of staining. You can stain a group of cells with different fluorescent dyes. A certain type of dye will only stain, let's say, a liver cell. Another type of dye will only stain, for example, a cardiac cell. So based on these dyes, you can actually get the cells to glow different colors. Okay. So when these cells are going through the detector, they'll actually give off these pulses of light when they pass through the detector. The laser is being held at the same point, but as the cells pass through this detection window, they give off a pulse of light. And those pulses of light can be detected by light detectors which are placed near the detector region. If you place a light detector here, this is called the forward scatter. Now this light, when the cell passes through, this light will get blocked, and the change in light can be detected by placing a light detector here. You could have a detector that's placed kind of off to the side, 45 degrees off the axis, and that's called side scatter. And the fluorescence is also placed in this region too. So there's actually three types of detectors. One is the forward scatter detector, the side scatter detector. The forward scatter is placed at 180 degrees, the side scatter is placed at 135 degrees. And the fluorescence one is also placed off axis. And the fluorescent one is also a light detector, but it's filtered to only detect a certain color of light. So in this case, the fluorescence detector is designed to detect only red light. So when you do flow cytometry, you end up getting these scatter graphs. Oops. So when the laser light passes, when the cell passes by this detection window, this light is diffracted, so it gives off side scattered light. These two on the side. It also gives off a forward scattered light. Now this results in draw this here. So let's say a cell is going through a detection window, and then you have APD stands for avalanche photodetector. It's a type of light detector. And you have the laser going in this way. So obviously when the cell is going by, the laser light will be blocked, and that means less light is going to reach the avalanche photodetector. So as a result, the signal that you'll see here versus time this is the detected light. You'll end up getting pulses like this. When the cell passes by, the light will drop down a little bit. Another cell passes by, the light will again drop down a little bit. So the amplitude of this is monitored. The width of this is monitored. Each pulse represents one cell going by the detector. So this would be, the amplitude of this would be considered the forward scatter signal. And you also have another detector that sits right here, which is a side scatter detector, and that detects how much light is bouncing off the cell and being detected here. So it's the scattered light. This is also an avalanche photodiode, but this is for side scatter, and this is forward scatter. The side scatter will also give, if we look at the side scatter signal versus time, it will also give you a signal, and the signal might look something like this, where it increases when the cell goes by there, because light is being refracted off the cell and going into the detector when the cell goes by. So this gives the side scatter amplitude. In flow cytometry, you can plot the the side scatter signal on one axis and a different parameter on the y-axis. For example, you could do forward scatter on the y-axis, side scatter on the x-axis, forward scatter on the x-axis, side scatter on the y-axis. Any two parameters you can choose, and to plot this on a scatter plot. Each one of these dots on the plot represents one cell. For example, if we look at this dot right here, this dot that means when this cell went by the detector, it had a side scatter signal of 2 times 10 to the 2, and it had a fluorescence signal of 2 times 10 to the 3. The fluorescence, I mentioned there's 3 types of detectors. The fluorescence is another type of detector. So these scatter plots, each one of these dots on the scatter plot represents one cell. If you have a mixture of cells coming into the system, then the cells will cluster into different groups. For example, you can see that there's a lot of dots right here because there's a population of cells that are approximately 0.98 microns in size. And you get different groups, different regions where there's a high concentration of dots, and that high concentration of dots means a certain type of cell population.
Would this be the same thing if you have a library to say that if you had this specific contaminant, it would be this intensity, it would absorb that much intensity?
If you had a specific contaminant?
Yeah.
Like in water, for example?
Right.
Yeah, so this could be applied to water analysis. Yeah, let's say you have a bunch of different particle sizes in your water sample. Let's say that you know that if the particle size is around 45 micrometers, then there's a very high probability that it's a certain type of bacteria, which might be dangerous. So you might look for a forward scatter signal that corresponds to a 45 micrometer particle. And so you would look at a certain region of the plot to see how many of those particles you have. So this can literally count the number of and types of particles. If you want to be more specific, you might put a dye, a fluorescent stain in your sample to dye the bacteria that you're looking for based on the bacteria's surface markers. Basically, then only that bacteria will glow a certain color, and all the rest of the cell types will not glow. So you get a fluorescence signal. In doing that, you can also count the number of bacteria that you have. So this is sort of an application. Now, a lot of the technologies is, how do we sort these particles by size? How do we focus them? So let's talk about some of those techniques used to do that. So hydrodynamic focusing, I mentioned already. So there's 2 streams kind of coming in from the outside. It could be coaxial too. If you have a small tube inside a larger tube, this focusing mechanism would still work. Okay, the middle stream gets squeezed by the two outer streams. So one limitation with this approach, just taking, let's say, two channels like this, this is called two-dimensional hydrodynamic focusing. This is because this is on a two-dimensional chip, right? You have three channels coming in, but the chip design itself it's a planar chip, right? So it's essentially a 2D channel. Now, when you squeeze, when you put in these two streams from either side, that squeezes the middle stream, but it squeezes it in the XY direction. Let's say this is the XY direction. Now, if you want a totally focused stream, you also have to squeeze it in the Z direction. Let me show you. There's no illustration here. You also have to squeeze it in the Z direction, meaning the direction that's into the page. Now, if you focus two streams like this, what will happen is you'll have a sheet, a curtain of the middle stream. So if we looked at the Z-axis, the middle stream occupies all of the Z-axis along the height of the channel. Right? We would like to squeeze it also in the height of the channel so that you only have one spot in the middle of the channel, which is where the particles are focused to. All right, so let me draw this just so we're totally clear. In a two-dimensional flow focusing, where you have flow coming in this way, flow coming in this way, the middle stream gets focused like this. Okay. However, if we were to look at the cross-section of the channel, we would find that the middle stream looks like this. If this is the middle stream. What we would like is to have something where it's squeezed in the Z direction as well. So it might be more like something like this. Squeeze in all directions. The reason why we want that is because when we do flow cytometry, when this laser is focused on one area of the channel, it's a very small region of the channel. That's called the detection window. Okay, and if the cells can be anywhere along the height of the channel, that's not good enough. You have to focus the cells down to a specific point within the channel cross-section. So you have to squeeze both in the X and Y. You also have to squeeze in the Z. So this is a simple technique to do that, which requires two channel layers. So this was called 3D hydrodynamic focusing using multi-channel layers. With the basic laminar flow, we can focus or squeeze particles in a single dimension. I'm sorry, this is what I was talking about in the previous slide. In these techniques, the particles are then focused into 3D. All right, so the concept here is that you first use a standard flow channel to squeeze in the X and Y direction, and then you have these two additional flow channels to squeeze in the Z direction. Okay, so if you have flow going from A to E, for example, if you have flow going this way, and you also have flow coming in B and C, so whatever's coming in B, let's see, where's a good picture of that? All right, let's just zoom in on this. So when we have flow coming in on A and then B and C like this, what will happen is that A will be pushed to the top of the channel because B and C are coming in below it. Okay, so this is going to be laminar flow in the Z direction. B and C are going to be coming in from the bottom and intersecting with A. And C is also going to do the same. So what happens is that at this point you'll have three stacked streams. A is on the top, then B, and then C is going to be all the way on the bottom. B kind of flows up this way, C flows just below it, and A flows at the top. So you have focusing in the Z direction by the time you get to here. Now, when you add the sheath flow in the Z along D, these two sheath flows, now you get focusing in the X and Y direction. So now you have both your particles would get focused to the middle of the channel like this. Another technique is called microfluidic drifting. The microfluidic drifting relies on a curved channel, and so there's some inertial forces that are experienced by the particles that cause them to aggregate into the middle, into a region at the middle of the channel. We'll talk a little bit more about these forces on these particles in the next few slides. So for right now, just assume that the main design aspect of this is a curved channel. In the curved channel, these particles experience some inertial forces, and those inertial forces result in these things called Dean flows, which ultimately brings these particles to the middle.
And this is all at laminar, right?
I'm sorry?
This is all at laminar flow.
This is all in the flow. The flow is laminar, but the particle Reynolds number can cause inertial forces to act on the particles. So we'll get into that. Yeah. Another way to do this is by putting chevron patterns at the bottom of the channel, the bottom and top of the channel, which essentially are doing the same thing. They're adding flows that cause the stream in the middle to be focused in both the Z direction and the X direction. So inertial particle ordering is what's called sheathless focusing, meaning where it doesn't require you to have a sheath flow, these flows coming in from the two sides. This technique, these set of techniques, relies on inertial forces. Okay, this is getting what Jeremy was talking about. At high flow rates, inertial forces on particles can be used to manipulate the particles in the flow. So the inertial forces dominate when the particle Reynolds number is greater than 1. So let's see here. First of all, so this is the particle Reynolds number. The particle Reynolds number is equal to the channel Reynolds number. This is the Reynolds number that we studied earlier, which is basically the flow velocity times the density of the fluid divided by the hydraulic diameter of the channel. So we know the Reynolds number already. The particle Reynolds number also takes into account the dimension of the particle in relation with the channel. So it's AP. This is the particle diameter, and DH is the hydraulic diameter of the channel. So it basically has to do with the dimension of the channel. So it's AP over DH, the ratio of the particle size versus the channel size squared. All right, so obviously the particle size can't be greater than the size of the channel, because then the channel would get clogged. But this ratio is squared and it's multiplied by the Reynolds number. So when you're operating at fast flow velocities, you can be operating at Reynolds number exceeding 1. Okay. Normally in microfluidic devices, or when you're operating in the creeping flow microfluidic devices, you're not using Reynolds number that high. But that doesn't mean you can't do it. And in fact, these techniques actually rely on a higher Reynolds number. So enough Reynolds number so that the particle Reynolds number ends up being greater than 1. Then inertial forces start to play a role. There's two types of inertial forces that act on the particles, the shear-induced lift force and the wall-induced lift force. So if we compare it to what we're talking about here, particle focusing relies on external streams of fluid to kind of squeeze the particles into a single flow line. That's the same thing that was done during flow cytometry. The fluid is squeezing the particles together. Now, the difference here is that the particles are self-organizing themselves in the flow. The particles themselves are self-organizing into certain places within the flow. So let's talk about what I mean here. There's two types of inertial forces. There's a shear-induced lift force. This is caused by a velocity gradient. This pushes particles away from the center line. So wherever the channel, this is a good diagram to look at. So in a typical channel, you'll end up with this parabolic flow profile. Velocity is fastest in the middle of the channel, and then it recedes as you go to the outer parts of the channel. So the shear-induced lift force pushes particles away from the center line. So this LS, FLS, is a shear-induced lift force, is pushing particles towards the wall. You have a second type of force called the wall-induced lift force, FLW. This is caused by the wake of a particle near the wall, and this pushes particles away from the wall. Okay, so this inertial force actually pushes the particle in the opposite direction. So you can see that there's two forces at work here. The wall-induced lift force is most significant near the walls of the channel. The shear-induced lift force becomes more significant as we go towards the center line. So at some point, the two forces are balanced, meaning that the shear-induced lift force is equal to the wall-induced lift force. At that point in the channel, which usually happens at around like 0.6, 60% of the channel diameter, the particles will basically have no forces that's either pushing them away or towards the center of the channel. In other words, they'll find an equilibrium position. That equilibrium position, there's some initial early work was done all in cylindrical channels, and that was found that the equilibrium positions are approximately 0.6 to 0.7 of the channel radius. In a rectangular channel, its situation becomes more interesting. There's actually more equilibrium positions. In this example, you can see that there's 4 different equilibrium positions where the shear-induced lift force is balanced by the wall-induced lift force. And those equilibrium positions depends on the cross-section of the channel. If you have a square cross-section channel, there's 4 equilibrium positions. If you start making the channel more tall and narrow, so a high aspect ratio channel, in those cases there's only 2 equilibrium positions. All right. But you see how this is giving you particle focusing for free. There's no sheath flow required, so you're just using inertial forces. So these types of things can be used to sort or focus cells. A little bit more about the inertial particle focusing mechanism. The shear-induced lift force moves particles down the velocity gradient away from the center channel. And near the channel wall, an asymmetric wake induced around the particles generates a wall-induced lift force. This is what I said in the previous slide. The two opposing forces balances at a certain location within the channel. So the particles settle at a distance about 0.2 times the channel diameter away from the channel wall. In other words, 0.7-ish from the channel center. So if you have a circular cross-section channel, in other words, a tube, the particles will organize in a ring at a particular radius within the channel. If you have square channels, then you can have these different equilibrium points. If you have a rectangular channel, you can have equilibrium points that look like this. And the particle Reynolds number is used to predict when a particle focuses. If you use a higher particle Reynolds number, the particles will organize faster. So this is an example where you can do sorting. Suppose at the beginning of the channel the particles are just a big mass or a big aggregate of particles, and then the inertial forces push the particles to certain regions within the channel. For example, in this case, it pushes them towards the end of the channel walls. And if you just have a bifurcation or something like this, then you can cause these particles to go off in one direction and the remainder of the flow in the other direction. So this is called an enrichment, because these two flows that are going out have a higher concentration of particles than what you started off with. Okay, and you could imagine that this type of technique could be cascaded. It could go through multiple stages for further enrichment. So you don't necessarily need straight channels. You can do inertial particle focusing in serpentine channels. In this example, there's a bunch of particles that go into the channel, and the serpentine channel focuses them down. So this migration here, this was some of the early work where some of the inertial particle focusing techniques were actually discovered. I saw an interesting talk on this by Dino De Carlo's group, where he was basically saying he discovered some of these things on accident. You know, as good scientists, you always observe. You always try to observe the results of your experiment and try to understand what's happening. So while he was a postdoc, he saw that they were trying to do blood analysis in microchannels, and they saw that the blood particles were actually, kind of self-organizing into streams like this, rather than being all in random places in the channel. And then later, it took them a while, but they eventually start to understand why it happened. So they did a lot of modeling. They looked at a lot of old papers on it, and this is what ultimately led to these slides that I'm showing you right now, the inertial focusing techniques. These were only really discovered and developed. The concepts were discovered back in the 50s and 60s, but they were applied to microfluidic devices, in the last 10 years or so. So this example shows that both symmetric and asymmetric channels can cause particles to focus into equilibrium positions, and you can get very, very fine focusing with the serpentine channel, 80 nanometer accuracy, meaning that the positional accuracy of the focus stream is 80 nanometers. All right, so these graphs sort of compare what the focusing happens in a straight channel versus an asymmetric curved channel and another asymmetric curved channel. These are all at different particle Reynolds number. So notice that in each one of these devices, at low particle Reynolds number, you don't see the focusing effect. You have to go to higher particle Reynolds number in order to see the focusing effects. This slide here, or this graph here, shows the fact that the particles are initially unfocused, but the more serpentine sections that it goes through, it becomes more and more focused. Presumably because these serpentine channels, the particles are being forced to turn direction, so there's inertial forces involved whenever there's a turn in the channel. So let's now talk about particle sorting on chip. As I mentioned, whether we're talking about environmental or life sciences applications, we'd like to be able to sort particles on cells on the basis of their size and other properties. So conventional technology generally uses membrane filters to separate them, or filter particles based on their size. And some of the drawbacks can be that the filters can clog. You can only discriminate between two sizes of particles. By the way, a filter type thing would be, just a mesh like this. Let's say you were to run your sample through this mesh. It has a certain mesh size, so the small particles will get through there, the larger particles won't. It's as simple as that. Right? One of the issues with this is that the mesh itself is going to capture large particles, so eventually the mesh is going to clog. And with the mesh, you can only really, with one mesh, you can differentiate between two particle sizes. One large, something that's larger than the mesh size, and something that goes right through. It's smaller than the mesh size. So it's kind of a binary type separation, two sizes of particles. So microfluidics, this is where microfluidics comes in. Microfluidic particle separators can provide a number of benefits. First of all, it provides continuous flow separation, meaning the particles are collected while fluid is flowing. and it can be run continuously. For in the example of the membrane filter, you can run it for a while, but then you have to clear off particles from the filter, put the filter back in, and then continue running it. With microfluidic particle separators, if they're designed properly, they can have continual input, and the particles will be separated on the basis of just their interactions with flow. You can also discriminate between multiple particle sizes, and you can also design particle separators based not just on size, but other properties as well. For example, like deformability or charge, dielectric properties. And if it's designed properly, clogging is not an issue. So let's look at some examples here. Pinch flow fractionation is one of the earlier papers that came out, and this was in about 11 years ago, 10 years ago. Pinch flow fractionation uses laminar flows to sort particles, and I think this is a very nice illustrative example. The idea behind pinch flow fractionation is that you have a liquid that has particles in it, and these particles could be of multiple sizes. And then you have another liquid coming in from this side, and this would be kind of like a buffer or a sheath. Now the role of the buffer or sheath is to push the particles against the upper wall of the pinched segment. So there's a pinched segment in the middle like this. Notice that the lower fluid is pushing the particles up against the wall. Okay, when the particles are pushed up against the wall, the larger particles have a center of mass that's farther away from the channel, and the small particles have a center of mass which is very close to the channel walls. At the end of the pinch segment, where the channel broadens out, it widens up at the end, the shear forces that act on these particles is going to cause the small particles to go off into the upper stream, and the large particles are going to be forced to follow a stream that's a little bit further down. The reason why is because you have laminar flows. If you have a laminar streamline here, that laminar streamline is going to continue this way, okay, and so on for all the laminar streamlines in the pinch segment. The main difference is where the center of mass is for the particle. The center of mass here is very close to the wall, so this is going to follow the streamlines very close to the wall. The center of mass for the larger particle is closer to the middle of the channel, so it's going to follow the streamlines more towards the middle of the channel. A very simple and elegant way to sort particles. They also show that you can separate or sort blood cells in this manner. Blood cells, by the way, they're not spherical particles, they're disks. But they showed that when they're in this pinch segment, the blood cells are actually, they all conform to this orientation when they're in the pinch segment. So they're able to sort in that manner. So these are examples where they sorted 1 micrometer particles from 2 micrometer particles in a very simple and relatively easy manner. It just uses laminar flow.
So they even orient. So they actually flip.
Yeah, in this case, this is interesting. This wasn't the main topic of the paper, but it was kind of interesting that they showed that disks will actually orient themselves according to the flow. Yeah. So this pinch flow fractionation concept can also be extended into a more general technique called hydrodynamic filtration. Hydrodynamic filtration relies on a series of branching channels. And in these branching channels, the particles are constantly being pushed towards the outer wall. So it takes that pinch flow fractionation concept and it's just repeating it over and over again, in some sense. That's the main kind of concept. But this is more specifics. Okay, it relies on a series of branching channels. So a branching channel, a branch point would look something like this, where you have the main channel here, and then there's two flows coming off the side. Excuse me. There's two flows kind of going off the side like this. Now, when you have flow out the sides, this is actually going to remove some of the fluid from the center channel. Every time you have one of these branching points where fluid is being pushed out the sides of the channel, this causes the particles to move towards the outer walls. Okay, so in a sense, we're pushing the particles up against the upper wall. We're also pushing them up against the lower wall. So this pinched flow fractionation type thing is happening on both sides of the channel. So when we're pulling fluid out of these branching sections, particles get pushed towards the sides, and eventually the small particles actually go out through these branch sections. Why? It's the same reason as before. First, okay, this is part B. Several particles are pinched against the wall. Some small particles exit out the branches. So this is what we're looking at here. The small particles have a center of mass which is very close to the wall, so the streamlines that they follow are very close to are the ones that will follow into this branch here. The larger particles follow this more the center streamline, so they kind of go straight here. Okay, the channels and the flow rates have to be designed depending on the size of the particles that you want to separate. So after passing more branching channels, a substantial fraction of the flow exits the main channel. At this point, large particles will also exit. Okay, so this is obviously not what you want. You want to be able to separate out the small particles from the large particles. But it allows you a way to just remove small particles from an agglomerate of more particles. Okay, so that covers that. This is a way for you to continuously remove small particles from a larger stream. And the second selection round here, you can see that there's mostly large particles, but there still might be some smaller particles in there as well. So these techniques are both just simply relying on laminar flows. It's relying on the interaction of particles with laminar flow. And the principle is that the particles will follow the streamlines where their center of mass is located. A very cool technique, which was published in Science magazine in 2004, is deterministic lateral displacement. Now, this is an interesting technique. You got to kind of look at it a few times to kind of get it. But it's a very highly efficient method, which is why it's worth understanding. This method is actually more efficient than pinched flow fractionation. But in some ways, the concept is still very similar. Particles are being pushed up against a wall, and then the separation and flow streamlines are causing particles to go in one direction or another based on their size. But check out this paper. I like it. It's a good read. So the deterministic lateral displacement is a highly efficient method which uses an array of staggered posts. And the beads that are small compared to the pillar spacing remain in their lanes, and they follow a straight path, whereas larger particles are deflected into neighboring lanes. So let's look at some of this diagram here. This is comparing the action of small particles versus large particles. Okay, deterministic lateral displacement relies on this array of pillars, and notice that the pillars are staggered like this. Okay, they're staggered in relation to the direction of flow. The flow vectors are going straight down like this, but the obstacles, if we look at a single line of posts, they're offset at a specific angle. So what happens is that the particle that goes down any streamline is always going to interact with the post underneath it. It's going to keep on interacting with posts as it goes through the channel. So let's compare what happens in a small particle versus a large one. The small particle, the center of mass is very close to the channel wall, just like we talked about earlier. Whereas with a large particle, the center of mass is farther away from the channel wall. In the case of the small particle, notice that it follows the streamline here. Okay, and the beads that are small, they're remaining in their lanes. They follow a relatively straight path. compared to the larger particles. Okay, so they're sort of remaining in the lanes here. Now these lanes are laminar streamlines. What happens to the larger particles? The larger particles are deflected by the posts. So notice that here, by the way, these numbers, I'm sorry, I should have mentioned them earlier. 1, 2, and 3, these are the laminar streamlines, the streamlines related to the flow. Now, like I said, the small particles, they follow whichever streamline they happen to come in on. But the large particles here, the large particle here is in streamline 2, right? The center of mass is in streamline 2. Streamline 2 would follow a path that looks something like this, and it would end up down here. Right? But the particle is deflected into the neighboring streamline because it's so large that it's displaced by the fact that this post is there. So the particle keeps on going off to the right, whereas the small particles will travel a more straight path. All right. You may have to look at this a few times to kind of really get a feel and intuition for this concept, but the key is that the posts are at an angle. Okay, and this works best with particles that are either smaller than the spacing between the posts or almost at the spacing of the posts. Now the advantage of this over previous techniques is that it has a very fine resolution. It can separate 0.8, 0.9, and 1 micron particles. So even particle sizes that are even differ by 10%, this one will be able to separate them. So it's really, really interesting, cool technique for separating particles by size. A follow-on work has actually shown that you can separate particles, separate cells by size as well. And these pillar arrays are being used for actually capturing cells now. The posts are functionalized with antibodies. I mean, that's a different topic here. This one, this on its own is a very interesting technique for separating particles based on size. The larger particles keep on being deflected, so they kind of go off to the right. The smaller particles follow a more straight path here. So going back to inertial, using inertial techniques. So these 3 methods of particle sorting all relied on just laminar flow, the interaction of particles with laminar flow, and in some cases the channel walls, in some cases the obstacles in the channel. Now, the Dean flow particle separators, which were investigated, I would say, a little bit later, in the mid-2000s. This was a paper published by 2008 by Ian Paputsky's group at the University of Cincinnati, which is a very well-cited paper, showed that you can use inertial methods to separate particles by size. Now, this had been demonstrated earlier with some of the work by Dino De Carlo's group, but this was an interesting method which used a spiral channel, and it was able to get very good separation resolution. So, the first question to ask is, why a spiral channel? Okay, if we go back to this technique, the inertial particle ordering, well, this was, the particles were already being kind of ordered or located at specific regions within the channel. But the problem, one of the issues with this is that look at where the particles are. Top, bottom, left, and right. So if the particles are at all 4 locations, there's no real good way where we could just take out one of these locations. We'd have to do multiple steps if we want to enrich the particles in this manner. What would be better is if we had all the particles being aggregated to a single equilibrium position. And so that's what the Dean flow things allow you to do. All the particles get aggregated to a single equilibrium position. All right, so just again, if you have laminar flow, if you have particles in laminar flow, the particles will be randomly distributed through the channel. If you include inertial forces, then the particles will form go to 4 equilibrium positions if the channel is straight. If the channel is curved, then the particles actually aggregate into a single equilibrium position. The reason why this works has to do with these circulating vortices called Dean flow. If you have a curved or spiral channel, the fluid is constantly going around in circles, so there's centrifugal forces. These centrifugal forces causes these rotating vortices to occur within the cross-section of the channel. So this is showing the curved section of the channel. Due to the centrifugal forces, there's a lot of force that's driving the fluid in the middle towards the outside of the channel. And this causes a co-rotating vortex. A vortex that's rotating counterclockwise at the top, it's rotating clockwise at the bottom. There's also the 2 inertial forces that we mentioned earlier, the inertial lift force and the wall-induced lift force. So there's the shear-induced lift force, the wall-induced lift force that we talked about earlier. The total force is the sum of the 2. And now we also have these Dean forces at work. The Dean forces just cause the particles to rotate along with these vortices. And the interaction of the inertial lift forces with the drag due to the Dean vortices, it causes the particles to aggregate into 1 equilibrium position at the inner wall. All right, and this is all governed by something called the Dean number, and they explored at what values of Dean number this phenomena occurs. The other thing they showed is that you can separate particles based on these flows. 7 micron particles versus 2 micron particles. Not quite the resolution of deterministic lateral displacement, but you can separate particles very rapidly in these devices because the flow rates are very high. These things are operating at in the inertial regime. So the separating particles is based on the concept that different particles will have different Dean numbers, and as a result, they'll settle at different rates into these inertial positions. So you can separate out these small particles from larger particles based on how quickly they equilibrate to their final resting point within the cross-section of the channel. So the second section here is moving on, is separation with active fields. Now, what all the previous techniques had in common, particle sorting techniques, is that they all relied on the interaction of the particles with the flow around it. So they were passive devices in the sense that you're not adding any electrical fields, you're not adding any vibrations into the system, you're not adding any energy other than just flowing the particles through the channel. That's one of the reasons why these techniques that we showed have been so popular. They're very simple to do. You just have to flow the particle sense suspension through a specific channel and you get these very cool and interesting sorting effects. But there's also a lot you can do with active fields. If you start adding electrical fields to it, acoustic fields, and so on, you can get interesting ways to separate cells. So we're going to talk about some of those. The concept of field flow fractionation uses external fields to sort particles on the basis of certain properties. So there's 3 methods in field flow fractionation. The general technique is as follows. Use some type of active field, like magnetic and electrical fields, to push the particles against a wall. Okay. Now this is similar in principle to pinch flow fractionation. The particles were being pushed up against a wall by the sheath flow. In this case, we're using external fields like electric or magnetic fields to push the particles against a wall. Once you do that, some interesting things happen. Let's look at the 3 things that can happen. Okay. In all 3 cases, we are assuming that the particles have been pushed up against the wall, the bottom wall here. In one case, suppose the particle sizes are very, very small. Now we haven't talked about this very much, but when you have particles that are very, very small, they are subject to something called Brownian motion. Meaning, Brownian motion basically describes the fact that everything in nature vibrates and moves. The only way to turn off Brownian motion is to bring the system down to absolute 0. and at that point Brownian motion would stop. Brownian motion means that all particles are just moving and vibrating all the time. So Brownian motion causes smaller particles to be further away from the wall on average than larger particles. If both of the particles are subject to Brownian motion, meaning they're very small, usually submicron, then you could imagine that the the particles that experience more Brownian motion are on average farther away from the wall, so they end up traveling through the system faster. Let me remind you of another important concept. Whichever particle is farther away from the wall will get through faster. Because remember, we have a parabolic flow profile in the channel. Anything that is closer to the center of the channel are experiencing the faster flow vectors. Anything that's very close to the channel walls are experiencing slow flow vectors. So particles will travel faster if they're closer to the center line of the channel. So in the case when you're in submicron particles and molecules, Brownian motion causes smaller particles to be further away from the channel wall on average, so they exit faster. In the steric mode, this is for particle sizes about half to 10 microns. Brownian motion plays less of a role, or doesn't play a role. And in this case, the center of mass of large particles is farther from the wall, so they exit faster. All right, so this is what we were talking about earlier. Larger particles have a center of mass that's, when they're both pushed up against the wall, the center of mass is further away from the channel wall. So the larger particles would travel faster through the channel than the smaller ones would. This is, there's no bifurcation in this case. All it is you can separate based on the fact that the larger particles travel faster than the smaller ones. Now, in this mode, the hyperlayer mode, as they call it, this is for particle sizes larger than 10 microns. When you have larger particle sizes, the hydrodynamic lift forces start to play a role. These inertial forces start to play a role. And these inertial forces move them away from the wall. Larger particles usually exit first, but the shape and deformability can also play a role. So in this case, large particles are exiting the channel before the smaller particles are. So, depending on which class or which regime you're working in, either the small particles or the large particles will come out first. So you have to understand what type of particle you're working with, whether it's subject to Brownian motion or hydrodynamic lift forces or just center of mass type laminar forces. Based on those methods, you can separate particles. Basically, these particles will travel through the channel at different speeds. Another method is this split thin flow fractionation. This is, I would suppose, more easy to understand. This is where you have a mixture of particles flowing through a channel like this. This is very similar to the H filter that we talked about earlier, except it's not relying on diffusion. The external field is pushing one type of particle to the second outlet, while another type of particle, which is less affected by the external field, flows through to outlet 1. So this is a field flow fractionation. Now, from a theoretical standpoint, there's all sorts of forces that you can exert on particles. Electrical and magnetic forces are just two examples. There's drag forces, there's inertial forces, and we've talked about those. There's sedimentation forces. Particles will sink to the bottom due to gravity. There's electrophoretic forces. Particles respond to an electric field. Electromagnetic force, the Lorentz force, we talked about when we were talking about magnetohydrodynamic pumps in the previous module. Magnetic forces, if the particle is magnetic, this is used throughout biology nowadays. They often use magnetic particles, and you can sort out the particles just by putting an external magnet on there. Dielectrophoresis is another type of electric field force. Acoustophoretic force, particles are actually attracted to the nodes and antinodes of standing acoustic waves. So if you create a resonating acoustic wave inside a channel, the particles will actually go towards certain points in the channel that represents the pressure nodes and antinodes. A very, very interesting technique. And then you can also use optical techniques. Interestingly, you could use light to manipulate particles as well. Light is an electromagnetic energy, and that electromagnetic energy can be can cause a particle to be deflected off in one direction or the other. So let's talk about some of those methods. First of all is gravity-based particle sorting. A simple and elegant way to separate particles based on size and density is to use gravity. So this is a channel. This was done by Shu's group at Michigan. Shu Takayama was my one of my faculty advisors when I was doing my thesis there. This is an elegant and technique that can separate particles based on their size. The key thing is that the microfluidic channel has to be placed on its side so that gravity is in the direction that you see here. So gravity vector is down this way. So the particles are all flowing through a channel like this. First, they're focused into a single file line like this. Okay, and at that point, then they enter the separation channel. The separation channel has an angle to it, so it's becoming wider at the outer end versus the inner end. This is called hydrodynamic amplification of gravity-based sorting. So laminar flows or drag forces are going to cause the particles to go from left to right. At the same time, gravity is going to cause the larger particles to sink to the bottom of the channel. Gravity is in this direction. So as a result, bigger particles will go towards the bottom outlet and smaller particles will remain at the upper outlet. And this is shown to be a very efficient method to separate particles by size. 3 micron versus 20 microns, 1 micron versus 20. These are diagrams showing the collection of particles in the upper versus the lower channel. So that's gravity being used. Gravity, if we look at this section of the diagram, sedimentation. The gravitational force scales as radius cubed. Okay. So the larger the particles are, the more gravitational forces they'll have. Since it's radius cubed, you can think about separating particles based on their size, because radius cubed means a small change in radius will result in a large change in the sedimentation force. Another example is to use magnetic forces. This is relatively straightforward. You can imagine that different particles could be magnetized, and magnetophoresis basically means you have flow in the channel. So in this case, the flow is going from right to left. You also have a magnetic field that's acting on the particles, and the magnetic field can cause the particles to migrate at different rates depending on their size and magnetic properties. So in this case, what happened? The sample mixture is going through the bottom streamline here. Okay, important point to note here. Oops. What happened? The sample is coming down the bottom part of the channel, and this is just buffer. Okay, this is like continuous free flow electrophoresis. If you don't have the magnetic field, all the particles will just travel straight through the bottom half. But if you include the magnetic field, the particles will travel, different size particles and different magnetized particles will travel at different speeds upwards depending on their properties. So the different types of particles would end up at different outlets here, and you could potentially count each one of these types of particles. One potential issue with magnetic particles is that the hysteresis can cause the particles to become agglomerated even after the field is removed. So let's say you have a cluster of particles here, they can start sticking to one another. This is the magnitude of the magnetic force, and notice that it scales as a volume, so it also scales as the radius cubed. So optical particle sorting. So first of all, the principle of optical tweezers. I mentioned that laser light, if you have a focused laser beam, that is that is a focused electromagnetic source of energy. So these electromagnetic forces, though they're not very strong, for very small particles they can be used to manipulate particles. If you're interested in this kind of thing, just look up optical tweezers on on Wikipedia or something. They were invented in the 80s, and they were invented in the 70s, although the technique has been perfected a lot since then. And they're used throughout physics and biology. They're basically using light to move stuff around. It's pretty cool. So the principle is a focused laser beam creates a strong electromagnetic field gradient near the narrowest part of the beam at the waist. A field gradient means that we talked about chemical gradients, so the electromagnetic field gradient is the same principle. The electromagnetic field is very strong right at the center of the waist, and then it drops very dramatically as you go towards the outside. And then once you're outside this region, you don't have an electromagnetic field at all. So there's a very strong field gradient, and it turns out that dielectric particles are very affected, or they are, there's a electromagnetic force that's exerted on a dielectric particle if it is within this electromagnetic field gradient. If a dielectric particle enters the beam, there will be a net force attracting it to the center of the beam. This is because the more intense portion of the beam transfers more optical momentum to the particle. So let's say that the particle is aligned to the side of the beam. This second vector here is exerting more force, more optical momentum than this one is. And that results in a net force that pushes the particle or pulls the particle towards the center of the beam. At this point, the forces are balanced. So once the particle is balanced, you can actually move that laser beam around and take the particle with it. Like I said, this is used throughout biology and physics. So you can use a laser for particle sorting. Basically, you can have a channel like this, and you focus a laser beam down here. You could have one optical detection window which kind of detects what type of particle you have, and then down here have the actual optical tweezer or laser which directs the particle into one outlet or the other depending on what you analyzed. For example, you might say, like, we're going to sort the fluorescent cells in one direction and non-fluorescent cells in the other direction. The nice thing about working in the optical domain is that it's very quick. I mean, you could turn on and off lasers at femtosecond speeds. So you can do this very, you can sort very, very rapidly. This was another example where they used an optical lattice. So we're not going to go into the details of that right now. But in this case, they actually didn't need to use a laser beam, but rather a pattern of optical light. But they were able to sort particles in that manner. And this was a famous paper that was published in Nature in 2003. Dielectrophoresis uses electric forces to separate particles. This is a fundamental phenomenon that was discovered by Pohl in the 1950s, and he wrote a book called Dielectrophoresis. If you expose a particle to a non-uniform electric field, then the particle will either get attracted to the high field region or it'll get deflected away from the high field region. So first of all, what is a non-uniform electric field? The way you get a non-uniform, well, let me give you an example of a uniform electric field first. A uniform electric field looks like this. You have two electrodes that are the same size. You put a voltage across it, and the electric field lines are the same everywhere between these two plates. Electric field lines are uniform. Now, if I were to take one of these electrodes and make it a different size, like this, if I make it smaller than the large one, now my electric field lines are going to look more like this. They still point from the positive to the negative electrode, but now they're not uniform anymore. So that's a non-uniform electric field. So whenever you have a non-uniform electric field, a dielectric particle, what will happen in the fact that you have a non-uniform electric field, you'll end up having a non-uniform charge distribution on the surface of the particle. And that non-uniform electric field interacting with the non-uniform charge distribution on the particle surface will cause the particle to move in a certain direction. So notice that this charge distribution on the particle it's actually an induced charge. It doesn't have to be a charged particle. It just has to be a dielectric particle. A dielectric particle is a particle that where you can induce charge on it by putting an electric field on it. The strength of the force and the direction of the force depends on the permittivity of the medium, the electrical permittivity of the liquids, and also the Clausius, which determines this thing called the Clausius-Mazzotti factor. This depends on the permittivity of the particle versus the medium. And the equation for it is given here. This is the dielectric constant of the medium minus the dielectric constant of the particle divided by EM plus 2 times EP. So depending on what the magnitudes of the dielectric constant are, you'll get either a positive DEP force where the particle is attracted to the high field regions, or you'll get a negative DEP force where the particle is attracted to the low field regions. So that's what these two are here. This positive dielectrophoresis happens when the Clausius-Mazzotti factor is greater than 0. The way you generate dielectrophoresis is that it generates non-uniform. You use non-uniform electrode geometries. Dielectric force is proportional to r cubed, so since there's a size factor in there, of course it can be used to separate or sort particles by size. In fact, this has been done, DEP-based sorting based on size. They used slanted DEP electrodes to provide DEP forces. Remember, to generate dielectrophoresis, all you need is some type of non-uniform electric field. And so people have come up with all sorts of ways to create non-uniform electric fields by different electrode geometries. So in this case, the cell types could be tagged with different size DEP particles, and this enabled cell sorting. They call this technique multi-target dielectrophoresis activated cell sorter, and this was shown to be able to enrich the cells by 100-fold. Okay, so this was an example of where DEP is used to sort based on size. This is another example. This is from Albert Folk's textbook. I'm trying to remember where the original work was. It may have been work by Peter Gascoigne's group, who's in Texas. He's done a lot of this work on the DEP dielectrophoresis. So they were showing here that you could actually use the magnitude of the DEP force. Remember that the DEP force scales as radius cubed. And the breast cancer cells have a larger size than the red blood cells. So as a result, at a specific electric field strength, there will be the breast cancer cells, which are larger in size, will have a larger DEP force, and they'll be attached to the electrodes. They'll get attracted to these high field regions, while the red blood cells just flow through. So it could be a way to separate cells based on size, and they show the application in cancer diagnostics. So acoustophoresis leverages acoustic fields to sort particles based on their interaction with the acoustic waves. So acoustic transducers such as PZTs can generate standing acoustic waves in a microchannel containing fluid. So particle in the fluid experiences an acoustic radiation force which moves it to the pressure node or the antinode. So when we think about standing waves, okay, these are called acoustic standing waves. Remember that the most intuitive example of a standing wave, in my opinion, is when you, let's say I were to take a slinky or a rope and to tie that end against the wall, and I'm just here moving this rope or slinky up and down. That's going to generate a standing wave pattern, and you guys know what that looks like. Certain areas will be going up and down like this, and other areas will be basically staying in the same place. You generate these things called standing waves, to draw what that looks like. So let's say my hand is here, and I'm moving this up and down, and this end is tethered to the wall like this. So this place is not moving, but this part moves up and down. You know, it does something like this. My hand is moving up and down like this. So this is called, these points here, they actually don't move. While these points, these 4 points, they're moving a lot. Okay, so this is an example of a standing wave. The places where they're not moving are called nodes, and the places where they're moving a lot are called antinodes. So it turns out that particles will be attracted, they'll move towards the nodes or towards the antinodes, depending on a number of factors. Rigid particles normally move to the node, while non-rigid particles or less dense particles move towards the antinodes. For like rigid particles will move towards the nodes, like things like air bubbles, which have a very low density, will move to the antinodes. So it's in a way it's analogous to dielectrophoresis. In dielectrophoresis, whether a particle is attracted to the high field region or pushed away from the high field region, it depends on the dielectric properties of the particle. In the case of acoustophoresis, it depends on the mechanical properties of the particle. So mechanical properties might include things like the compressibility, beta. It may include things like the density of the particle. Okay? And it also includes the particle size as well, just like the other techniques. So, you know, it's interesting that you can get particles to move to nodes or antinodes based on their density, for example, whereas dielectrophoresis is good for sorting based on their electrical permittivity. So free flow acoustophoresis exploits the fact that the forces are size dependent, so this could be used to sort particles based on size. A lot of this work, it's some really nice work by Professor Thomas Laurel's group at Lund University, where they showed, you know, that these particles will actually go to these nodes and antinodes, and that can be used for separation. And there's a very good theoretical basis for this. In this example, they showed that particles will migrate towards the nodes or antinodes at different rates, and so you can leverage that fact. So you just have a small region where this where the transducer is mounted. Just in that region, you'll get this acoustophoretic effect, and you can design it in a manner such that they start to migrate, and then once they get past this region, they stop migrating. So you could have them separated out at this point in time, based on the rate at which they migrate. So these red particles move very rapidly towards the center, so they make it all the way to the center. But the smaller particles, which migrate slower, they don't make it, so they end up towards the outer streamlines. And you can have several outlets in this way. So a very interesting technique. This technique has picked up a lot of interest, especially in the last 5 to 6 years. So the last technique I'll talk about today is single cell trapping. So in many biological studies, we would like to perform tests on biological cells. Now this topic I included as a part of this module because I think it's a logical extension to some of the techniques that we saw earlier. This is not a sorting technique, but it is a way to trap cells and particles once they have been sorted. Or they could also be trapped in unsorted format. So let's just think about this from the life scientist's perspective. So in many biological studies, we'd like to perform tests or assays on biological cells, and several common themes emerge. So how can we isolate cells from solution? You know, and use them, for example, we'd want to use them for blood analysis. How can we sort cells based on cell type? This would be used in flow cytometry. How can we create arrays of cells? These would be useful for high throughput studies of cell biology. So there's a bunch of different examples of single cell traps that I would like to go over, but I just realized that we have 10 minutes left in class, and I wanted to do the course evaluations today because I don't think that we'll have time for that. I don't think that we'll have time for it next week because we'll be busy with the presentations. So I'm going to give you guys an option. Let me know which one you prefer. We can do the course evaluations next Monday after the first set of presentations, but it may require us to stay an extra 15 to 20 minutes in class, so class would end at around 7:40 next Monday. The other option is, and then I'd spend the next 10 minutes really covering single cell traps. The other thing we could do is I could leave this. I could just record it and you can watch it later. We can do the course evaluations right now. Next Monday we can just do the presentations and then just end class at normal time. So how many folks would like to, how many folks prefer the second option, which is just to not do this right now and to do the course evaluations now? And the first option is we'll do the course evaluations next week, but it might mean that class is going to run over a little bit next Monday. So how about doing the course evaluations now? How many people would like to do the course evaluations now? Okay, 1, 2, 3, 4, 5. Okay. And how many folks would like to, okay. All right, so it's 5 versus 3, so I guess we have a majority. Let's do this now. I will pass out the evaluations right now, and I'm going to record some of this for your later interest online. I think it's an interesting topic, so please do look over this later. It's a way that you can create single cell traps for studying large populations of cells one at a time. Okay, so we'll do that. Let's end here.